ready.
Cool.
Here we go. Kia ora koutou, ko Douglas Walker, toko ingoa. I'm here today with Amy Rowe. Amy's a teacher at Wellington Girls College, and Amy's going to take us through the NCEA Level 2 Biology Externals. We're starting today with cell processes, and it's great to see a number of you already on. If you'd like to type any questions in chat as we go, I will either feed those to Amy as we're going, or I'll collate them and ask at the end, just whatever seems appropriate at the time. And I'll also put in a wee link for Study It. It's in the description. If you've never used Study It before, it's a forum where you can go and get help with any exam questions from teachers across the country. And there's a wee survey that you could fill in to help them improve the facilities of the forum. And that should only take 2 minutes. So without further ado, over to you, Amy. Thank you.
Kia ora koutou. It's great to see you all here. Hopefully, I'll be able to refresh your memory about some of those topics that you have studied in your 12 bio classes this year. So I'm just going to share my screen. Okay, so we're starting off with life processes at the cellular level. I think this is a fantastic place to start because it does provide a really good foundation for all of the material in the year 12 course, really. And as we know, cells are the building blocks of life, so we should probably know how they work. Oh, hang on, what am I doing? Right, here we go. So life processes at the cellular level, different textbooks seem to break this topic up into various different ways. There is a tendency to break it up into 3 chunks, but for me, I think it breaks up more cleanly into a few more. So this will be a little bit of a roadmap of where we're going with the topics that we're going to cover off today. So we're going to quickly look at an intro to the cell. Just a reminder, all of that should be nice and easy for you. From there, we're going to look at the cell membrane, because that is the first, if we were going to take a journey through a cell, the first thing we would encounter as we come up to the cell is the cell membrane. It is a very vital part of the cell, and it influences a lot of the life processes from there. So we've talked about the cell membrane. We're going to be looking at cell transport. So just a reminder, we'll be talking about passive transport and active transport, and there's different headings within those. Well, we're going to do a rundown on proteins and enzymes, talk about the importance of protein structure, the role of proteins and enzymes in the cell, what it means to denature an enzyme, and look at those factors affecting the enzymes. Now, when we talk about the factors affecting the enzymes, that is pretty important. Because those factors that are affecting our enzymes will be affecting the other life processes that we're going to be talking about, such as respiration, photosynthesis, and DNA and mitosis, or DNA replication and mitosis. All right, let's start with an introduction to our cells. Okay, so the level 2 material looks at eukaryotic cells. These are our animal cells and our plant cells. We don't look at things like bacteria in any real detail. So here we have our animal cell and our plant cell. Around the outside of our animal cell, we have our plasma membrane. Inside, holding all of our genetic material, is the nucleus. And you've probably heard of a number of these other organelles, the smaller organs within inside the cell. So we have our mitochondria here. You may have heard of the Golgi apparatus, vacuoles, and endoplasmic reticulum, and hopefully you've heard of ribosomes as well. Plant cells are reasonably similar. They do have a plasma membrane around the outside, okay, but outside of our plasma membrane we have a cell wall, which is a tough cellulose-based rigid structure that goes all the way around the outside of our plant cells. It's important for the structure of plants because it helps give them shape and structure and helps hold them in position. Unlike animals, plants don't have a skeleton, so they do rely on their cell wall to help give them structure. Very similarly to our animal cells, we have a lot of the same organelles like the endoplasmic reticulum, ribosomes, the nucleus, and all the rest of it. But unique to our plant cells is our chloroplasts. So we're going to be looking at those in detail later. All right, here is a summary. First off, we have our plasma membrane, controls the movement of our water, gases, and other substances in and out of the cell, and it provides the outside coating of the cell. And both of our animal cells and our plant cells have those. Our cell wall gives our plant cell structure and rigidity, which we'll only find in plants. Shapes, okay, because of the cell wall, our plant cells tend to be more rectangular or cuboidal or square in shape because of that cell wall. Animal cells aren't bound by that cell wall, and so often animal cells can have a wide variety of different shapes, often rounded. And their shape is closely correlated with their function in the body as well. Inside of our plasma membrane, we have the cytoplasm. So this is a jelly-like fluid in which all of the cell's contents float. So all of the cell's organelles will float in there, as well as metabolites and enzymes and gases and other liquids and foods for the cell. So it's kind of like the juice inside the big gooey cell. A nucleus is what separates and protects the nuclear material from the rest of the cell contents and helps protect it from enzymatic degradation from things inside the cell in the cytoplasm. Our ribosomes, we'll talk about that more in the gene expression topic, but these are the site of protein synthesis, so they're very important. And then lastly, our mitochondria and chloroplasts. So mitochondria are found both in animal cells and plant cells, and they're responsible for generating energy in the form of ATP. Whereas chloroplasts are responsible for photosynthesis. So they take energy from the sun, they take water, they take carbon dioxide, and they use those to make glucose for our cells, for our plant cell. Let's have a look at the plasma membrane. It is the outside layer of our animal cell, and it's one in from the cell wall in our plant cell. It's responsible for keeping the cytoplasm of the cell and all of the organelles contained in one place. It controls the movement of water, ions, gases, and other substances in and out of the cell. Okay, so it's got a very, very important role. The plasma membrane is what we call a phospholipid bilayer. It has two layers of phospholipids. We talk about phospholipids. We've got one here on the left-hand side. They're always drawn as these sort of closed peg-type shapes. It has a glycerol phosphate head, this one here, okay, and hanging off of that are two fatty acid tails, and fatty acids are also known as lipids. So where we get the name phospholipid bilayer or the phospholipid, the first part is from the phosphate head, and the lipid is the fatty acid tails. Now, our phosphate head is hydrophilic. That means that it loves water. It will bind to water, and it will bind to water. And then we have our fatty acid tails down here, and we say that they are hydrophobic. So they are water-fearing. They will not mix well with water. Now, the cell in most of the environments that cells are found in, there's a lot of water around. Okay, so when we get a whole lot of these phospholipids grouping together, they take this bilayer construction or shape over here on the right-hand side. So you can see that there are lots of these clothespin-shaped units with our phosphate heads up here and our fatty acid tails down here. And on the top here, they're all sort of lined up with their phosphate heads together. Okay. However, it would be fine if there was a whole lot of water extracellularly out here, okay, but not intracellularly inside the cell. So we need to protect these fatty acid tails, okay, which are hydrophobic. They don't want anything to do with water. So the way the cell gets around this is it provides another layer of phospholipids. So we have these two layers, the top one here and then the bottom one here. Our fatty acid tails point together with our phosphate heads on the inside and the outside of the cell. This means that water and water-soluble solutes on the outside of the cell and on the inside of the cell can freely move around, and we don't need to worry too much about those fatty acid tails being affected. So here is a zoomed out view of our plasma membrane. You can see we have our phospholipid bilayer here. We've got our phosphate heads all lined up, and we have top and bottom, and we have our fatty acid tails all congregating together in the middle. Throughout our membrane, we have these integral proteins, or proteins that are stuck inside our phospholipids, and they're kind of scattered like a mosaic. So we call the plasma membrane a fluid mosaic. So fluid because all of these proteins and extra features in the cell are free to move around. Okay, they sort of move around through our phospholipid bilayer, kind of like icebergs floating on the sea. Okay, they're free to move around. And we say that it's a mosaic because there are many different pieces scattered throughout our cell membrane, or many different features. So we have our protein channels, these ones here. Okay, we have these other integral proteins that span from one side of the membrane to the other, and they have carbohydrate chains attached. More about them in a minute. And we have lots of other proteins that perform vital functions to our membrane. So we say that the membrane is semi-permeable and selectively permeable. So semi-permeable means that not everything can get through it. It will allow some things through, very small molecules, and uncharged molecules are able to pass right through the lipid bilayer. And it is selectively permeable because it does restrict the movement or control the movement of different molecules. And that's where we're going to talk about transport across the membrane. So some small molecules can pass through the membrane, but others can't. Protein channels, like these ones here, and carrier proteins help to control which molecules can move into and out of the cell. So here is our diagram of our plasma membrane again. I've made it a little bit smaller there. Some other features that you will see in our plasma membrane, you might hear of cholesterol. This is a molecule that helps to stabilize the cell membrane, reduces its permeability to water-soluble molecules, so they're not constantly rushing backwards and forwards, and helps to stabilize the membrane. We will have protein channels and protein pumps, these ones here. Okay, these allow the movement of specific molecules in or out of the cell, and we'll be looking at those in greater detail soon. You may hear about glycoproteins. These are often receptors for specific molecules or neurotransmitters if we're looking at nerve cells, and they trigger cellular responses when these molecules are detected. So they are able to detect specific molecules, and then the cell responds by doing something once those molecules are detected. And then lastly, these blue chains hanging off some of our proteins here are what we call carbohydrates or oligosaccharides, and they're involved with cells recognizing self-cells, so cells within the same body, and they're able to signal between different types of cells. The most important ones that we need to look at are our channel proteins and our carrier proteins. So we're going to talk about cell transport. This is how cells move substances into or out of the cell or through the membrane. Okay, we're going to look at two types: passive transport. This is a type that doesn't need ATP or energy, and it moves substances along the concentration gradient. So they move from an area of high concentration to an area of low concentration. If you don't remember what that means, don't worry, I'm going to go into that in a bit more detail. Types of passive transport that we talk about are diffusion or simple diffusion. That's this one up here, where we see very small uncharged molecules being able to pass directly through the plasma membrane. We're also going to talk about facilitated diffusion. So this is where our molecules need a little bit of help to get through the plasma membrane. They're still going to move along the concentration gradient, but they will need some sort of channel to pass through the membrane. And then we're going to look at a special type of passive transport, which is osmosis. It deals with the movement of water. The other type of cell transport is active transport. So this is the type of transport that does require energy. Okay, we need some ATP to power the process. And the reason that this process needs energy is because we're going to be moving our molecules against the concentration gradient. So we're going to be looking at things like protein pumps, endocytosis, and exocytosis. You can see in this diagram here, we've got these little green squares. They're being moved through the plasma membrane from an area of low density to an area of high density. So we need some energy to make that happen. All right, so diffusion. It's a reminder diagram up the top here. Particles will randomly spread out from an area of high concentration, as we see on the outside of our cell here, to an area of low concentration. Okay, there are much less brown circles inside our cells, so these tiny molecules are able to spread through the cell membrane until they reach an equilibrium, until they're all spread out. Okay, so a concentration gradient, I've drawn these, or I've got these little blue dots here, and you can see that there is a concentration gradient of these blue dots across the bottom of my slide. So on the right-hand side, I have a high concentration of my blue dots, and on the left-hand side, there is a very low concentration of blue dots. And in the case of diffusion, my blue dots would spread out and move from an area of high concentration on the right here, and they would spread out all the way across until the blue dots were evenly spaced across the width of the slide. So there are a couple of things that affect the rate of diffusion. The first one is the difference between a high and low concentrations. So if there is a very high concentration on my right here and a very low concentration on my left, those blue dots are going to spread out and move down that concentration gradient reasonably quickly. Okay, they're going to spread out and move away from this area of high density. The size of the particles matter. The small particles will diffuse faster than large ones. And the temperature also matters. So if you remember from your acids and bases topic in year 11, if you did it, we know that an increase in temperature causes particles to move around faster with more kinetic energy. So if we increase the temperature of our particles, they're going to move around faster, they're going to diffuse faster away from this area of high density. The state matters. Okay, so we know that gases will diffuse faster than liquids. Again, it comes down to temperature and the size of the particles. Small uncharged molecules, things like oxygen, carbon dioxide, glucose, are able to freely pass through the plasma membrane. Okay, and they do so by moving down a concentration gradient, so from an area of high concentration to an area of low concentration. An example of this would be if there was a low amount of carbon dioxide inside our cell, but outside the cell there was a lot of carbon dioxide, we would have our carbon dioxide molecules easily diffusing, passing through our membrane to the inside of our cell. And that would continue randomly until there was an even concentration or equal concentration of carbon dioxide inside the cell with outside the cell. Okay, so not all molecules, in fact, most molecules are not able to easily diffuse through our plasma membrane. They need a little bit of help, and that's where facilitated diffusion comes into play. So we can see in our diagram here, these spiky little orange triangles are not able to pass directly through the plasma membrane of our cell, but they can pass through this channel. So they need a protein channel in order to pass into the cell. Many ions or molecules aren't able to diffuse directly through the membrane because they're simply too big, so they're not able to pass between the phospholipids, or they carry a charge, and the charge then interferes with our fatty acid tails with the bottom of our phospholipids. Okay, so if they're not able to simply diffuse through the cell, they're going to need a protein channel or a pore through the membrane to enter the cell. We call these transport proteins. They are specific. This means that they only allow certain molecules to pass through. They transport large molecules or charged molecules, ions, and other things with a charge. So, for instance, sodium ions and potassium ions. The particles still move at random, okay, and from an area of high concentration to an area of low concentration. But instead of going directly through the membrane, they need to find a channel. They need to find a door to go through. So, in this little animation that I've got from the amoeba sisters, you can see we have our particles outside the cell. In this case, we have passive transport. They are diffusing through the cell using diffusion. And on the right-hand side, we have facilitated diffusion. Our little molecules need to pass through our protein channel to get inside the cell. Okay, they are moving from an area, in this direction, they're moving from an area of high concentration to an area of low concentration. The important thing about both of these processes is they happen passively. There is no energy required, so no ATP is used. Excuse me. All right, so let's talk about osmosis. This is a special type of diffusion. It does tend to confuse people. So when we're talking about osmosis, it is a passive process, so there is no ATP used, and it is a type of diffusion, but it's the movement of water. And that's where it gets a little bit complicated because people tend to think about the molecules and the ions and the things in the water a lot, and don't tend to think about the water. So the official definition is that osmosis is the movement of water from an area of high water potential to an area of low water potential, which sounds a bit complicated. Okay, so when we talk about a solution having a high water potential, it means that it has lots of water and not much else. Okay, so solutes, these are the particles or the molecules that are dissolved in water. In a high water potential solution, we would have a low amount of solutes, a low amount of salts or anything else dissolved in it. It is mostly water. If we have an area of low water potential, this means that there's a lot of other stuff in it. Okay, so a lot of solutes. So we can see on this side we've got a lot of these green triangles taking up a lot of the space, and not so much of the water. Okay, we say that there is a low water potential. So osmosis is the movement of water or the diffusion of water. We have in the middle of these two solutions here this semi-permeable membrane. So a semi-permeable membrane won't let our triangles pass through. Okay, we don't have a normal diffusion process, but it will let our water pass through our membrane. Okay, so we see that our water molecules will move from an area of high water potential on the left here. They're able to pass through our semi-permeable membrane into an area of low water potential. Okay, the solute's not able to pass through because it can't fit through the membrane, but the water can flow. And this water flow will happen randomly and passively until we reach an equilibrium, until both sides of our membrane have a similar water potential. This is a really important process for the cell. Okay, so as I said before, cells are surrounded by water. They've got a lot of it on the inside of the cell and they've got a lot of it outside of the cell. So they need to be able to control the movement of water across the membrane. And this is called osmoregulation. The concentration of solutes, salts, and other molecules inside of our cell, in this case, is higher than outside the cell. Okay, we've got all of these solutes, our green triangles are our solutes, and we can see that the concentration of them inside the cell is much higher than outside of the cell. We would say that this solution out here is hypotonic. This means that there is a low amount of salt or a low amount of solute compared to inside. Excuse me. Then we put our cell in a solution with a lower solute concentration, as we've done here. The outside of our cell has a higher water potential, and the inside of our cell has a lower water potential. What this means is that our water on the outside here is going to move to where there is a lower water potential or a lower water concentration. So all of our water here is going to try and rush into our cell. Okay, our water is going to move from an area of higher water potential to an area of lower water potential inside the cell. This may have a couple of different consequences for our cell. Okay, if it's an animal cell, it will lyse. Okay, it will pop basically. So the water will continue rushing into the cell until the cell membrane cannot hold the cell together any longer. And we see that in these red blood cells on the top right-hand corner. So they have lysed, they have popped. Disastrous. However, if you're a plant cell and you have an increase in the amount of water rushing into your cell, it might be a good situation, a happy situation for you. Okay, and the difference is that plant cells have a cell wall all the way around them. Okay, when the water osmoses into the cell or passes into the cell, it's stored in a big vacuole, okay, which is a big storage vessel, and that vacuole fills up with water and it presses against the outside edges of our cell wall. Okay, this creates a lot of pressure, and we call that turgor pressure. We say that the cells are turgid, and for our plant cells, that's a really good thing because turgor pressure helps keep our plant stems upright and strong and functioning as they should. It helps give them structure. When a plant cell starts to lose turgor pressure, the plant starts to wilt. So you've all, I'm sure most of you have seen picked or cut flowers before. The first thing you do is you put them in a vase of water. And that's because those plant cells will continue to take up the water, packaging it into their vacuole and keeping those plant cells turgid so that your flowers stand upright in the vase. Okay, if they start to lose water, if you forget to pop up the vase and they run out of water, those plant cells will lose water and become flaccid, and that's when we see the plant stems starting to fold over and to wilt and droop. Okay, so in this case here, we have a concentration of solutes inside the cell, and here, okay, our little triangles, is much less than outside the cell, which is just packed with salts or solutes. Okay, we say that this solution is hypertonic, so high amount of salt. When a cell is put into a solution with a higher solute concentration, as we see here, it's experiencing a lower water potential. So there is a high water potential inside the cell and a lower water potential outside the cell. And we know with osmosis that our water will move from an area of high water potential inside the cell to an area of low water potential outside the cell. Okay, so our cell is going to lose water. It's going to become dehydrated. In the top right-hand corner here, you can see our red blood cell again has lost water and it has shriveled up. So instead of being a nice plump round red blood cell, it's sort of shrunk away and it's lost a lot of its shape and kind of looks a little bit like Australia. So it has shriveled up. And again, animal cells are able to do that because they don't have a cell wall. Still reasonably catastrophic for our cell. If it becomes too dehydrated, it will lose function and die. On the other hand, down the bottom here, we have our plant cells. We say that it has become plasmolysed. Okay, so our plant cell has lost all of its water from its vacuole, and you can see that it's taken this weird star shape. Okay, and what has happened is it has lost so much water that the inside has shriveled or shrunk, and it has pulled away from the inside surfaces of our cell wall. We see it in this weird star shape. The vacuole is completely empty of water now, and the cell inside our cell wall has shriveled, and the plant itself will have become droopy and flaccid. So it's got no water, it's got no turgor pressure there to help hold it upright. All right, so if we have a situation where the concentration of solutes inside the cell is the same as the concentration of solutes outside the cell, we say that this solution is isotonic. So this is an ideal situation for our cell. There is an equal concentration or an equal potential of water both inside and outside the cell. So there is no net movement. Okay, there's not a whole lot of water rushing into or out of the cell. However, water is still passing into and out of the cell, but it happens randomly. Okay, the water potential or the difference between the inside and the outside of the cell remains the same. Okay, so the concentration of the water and the solutes stays the same. It's a really happy place for our cell to be. No risk of dehydration, no risk of bursting. This is an ideal situation for our cell. And if the cell is part of a body structure, then this is the environment that it wants to maintain. Okay. So here is a wee question from a past exam paper, which references osmosis. So our lugworm is a marine creature here. It lives on sandy shores, and it is subject to changes in salt water concentration. So just with the tides, sometimes the salt water will be more salty than others. To survive in this habitat, the lugworm will passively adjust the salt water concentration of its body so that it matches the surrounding seawater. Oxygen consumption remains constant during this process. Let's not worry about that. The question says, explain how salt water moves across the cell in a lugworm via osmosis and facilitated diffusion. Okay, so first things to note, if we look at the question says that the lugworm passively adjusts to the salt water concentration. So we're not talking about ATP, we are talking about passive transport. And it also says that the saltwater concentration of its body matches the surrounding seawater. Okay, so this means that if the seawater increases in salinity or saltiness, then the environment inside our lugworm's cells also needs to increase in saltiness. If the saltwater drops salinity, so the concentration of salts becomes lower, then the concentration of salts inside our lugworm needs to become lower as well. We're asked to mention two processes. One is osmosis, and the other one is facilitated diffusion. Well, osmosis is going to be talking about water. So we know that water is going to be moving in or out of our lugworm's cells, and the other one is facilitated diffusion. Okay, so facilitated diffusion is going to be how the salt molecules move in or out of our lugworm's cell. When the salinity of the seawater increases, okay, somehow our lugworm needs to get more salt molecules inside of its cells. Okay, so that's where facilitated diffusion will come in. If the salinity of the seawater decreases, so there's a higher water potential, then the lugworm is going to need more water inside its cells. So it's going to need to use osmosis to have water flow from outside the cell to inside the cell. Let's have a look. So water moves across the lugworm's membranes by osmosis, and salts move by facilitated diffusion. The salt is moving from an area of high concentration in the seawater to an area of lower concentration inside the worm. Okay, this is with the concentration gradient. Sorry, I can't see the right-hand side of my screen because there's a wee window there. Let me see if I can. There we go. Hide that. So our salt is moving from an area of high concentration in the seawater to an area of low concentration inside the worm, down the concentration gradient through the protein channels in the cell membrane. Water, on the other hand, is moving from an area of higher water concentration inside the worm to an area of lower water concentration in the seawater, and it diffuses straight through the membrane. So our salts require facilitated transport. It requires a protein channel, whereas water can diffuse straight through the membrane. Osmosis and facilitated diffusion require no energy. They are both passive transport, and they work together to ensure that the lugworm always has the same saltwater concentration as the surrounding seawater. Right, so let's have a look at active transport. Moving molecules around across the cell membrane is not always so easy. So active transport uses energy to move molecules against a concentration gradient. So this is moving molecules from an area where there is a low concentration of them, or there's not many of them, to an area where there's already a lot of them. Okay, and it's kind of like pushing a car uphill. It needs a lot of energy. And the way that cells use energy is through ATP, which is adenosine triphosphate. So the tri says that we have an adenosine molecule with 3 phosphate groups attached. When ATP is converted to ADP, which is adenosine diphosphate, adenosine with 2 phosphate groups attached, this reaction releases energy. Okay, this energy is then used to power protein pumps, to move molecules across the membrane, to change the shape of the membrane in some cases, and the cell can use this energy to perform its function. So here on the right-hand side, we see our cycle. Here is our adenosine triphosphate up the top. It's got 1, 2, 3 phosphate groups. When one of those phosphate groups is cleaved off, it releases energy for the cell to accomplish its tasks, and it releases heat as well. And in response, we get ADP as the product. Okay, so adenosine diphosphate, only 2 phosphate groups. It's kind of like thinking of ATP as a full battery and ADP as our flat battery. However, the cell does have processes that will recharge the ADP by adding a phosphate group into it. The energy is absorbed, and we get more ATP produced, which stores our energy for us. So back to active transport. Active transport is selective. Okay, it moves each carrier protein moves specific molecules from an area of low concentration to an area of high concentration. Active transport is performed by ion pumps and carrier channels in the cell membrane. And as I said, only specific substances can bind to our carrier protein. Okay, so here on the right-hand side, we have our carrier protein. We have inside the cell on the bottom and outside of the cell at the top. And we can see that our little purple substances, our molecules that need to be transported, are able to enter and bind to our carrier protein. Importantly, ATP also binds to our carrier protein, and our carrier protein is able to cleave one of those phosphate molecules and turn ATP into ADP. When this happens, the molecule gets a shot of energy. This energy that's released is used to change the shape of our carrier protein. So you can see on the left, we have our carrier protein is open to the inside of the cell. Once that energy is released, it changes the shape of our carrier protein. So it closes on the inside and opens up a channel on the outside of the cell. And what this means is that our molecules that are being transported are now able to be released outside of the cell. The ATP is needed for the carrier protein to change shape, which ejects the substance to the other side of the membrane. Again, this is used in situations like absorption of glucose in kidney tubules or for moving charged ions around. Once again, it moves substances against the concentration gradient and it requires ATP. Those are the key points of active transport. So let's have a look at our potassium ion pump. This is an example of a carrier protein that is able to move charged particles, in this case our sodium ions and our potassium ions, from one side of the cell to the other. And these sodium-potassium pumps are important for the normal functioning of nerve cells and muscle cells. They're effective. What they do is they move 3 sodium ions out of the cell and bring 2 potassium ions into the cell each cycle, and they use ATP for energy. So over here in our first diagram, you can see our carrier protein, our pump here, and it has these 3 little sites where our sodium ions can bind. When those sodium ions bind and they fill up, and ATP is able to bind and be converted to ADP, what happens is it leaves behind a little phosphate group, okay, which is now bound to our carrier protein. The energy released from ATP and the phosphate group attached causes our pump to change shape. So it's kind of like a little bit like a revolving door. Okay, this bottom end of our pump closes and the top end opens. Okay, and what happens now is that our sodium can now be released outside of the cell. Our pump is now open to the outside of the cell where we see potassium ions. So our potassium ions are now able to come in and bind to potassium-specific sites on the inside of our carrier protein. When this happens, once our potassium ion spots are full, our phosphate group is able to detach from the pump, which causes our pump to return to its original shape. Okay, and its original shape is open to the inside of the cell. When this happens, our potassium ions are able to be released from our protein pump and into the cell. Okay, so we have this active process which requires ATP, and we see the net movement of 3 sodium ions moving to the outside of the cell and 2 potassium ions moving to the inside of the cell with each cycle. So another type of active transport is cytosis. Rather than looking at the singular protein carrier level, these are looking at large amounts of material and how the plasma membrane allows those large amounts of material to move into or out of a cell. And this happens by folding of our plasma membrane. We talked about the plasma membrane being a fluid bilayer, which means it is able to change shape when required. However, if it is changing shape, it requires a lot of ATP energy. So the first process we'll look at is endocytosis. This is the process by which substances are taken into the cell. So in this case, on the exterior of our cell, we have these little purple dots, and you can see they are being packaged with part of the cell membrane into these little things called vesicles. Okay, so they are membrane bound, they have a membrane all the way around them, and our particles, our molecules, are stored inside the vesicle. And we have a little animation on the left-hand side here. We can see it happening in real time. The way this happens is that the cell membrane folds in on itself. We say that it invaginates, so it creates a bit of a dip. Our molecules or material moves into that dip, that invagination, and then the two sides of our plasma membrane then come together over the top of that little hollow. And where they come together, the plasma membrane is able to heal itself. Okay, so you can see that that's what's happened here. Our plasma membrane has healed itself. Those phospholipids have sort of melded together, and the phospholipids, or the part of the membrane that was circling the materials, then melds together at the top and links together. So it creates this one unbroken loop of phospholipids with our materials on the inside. So this is the way that cells are able to take in solid particles or solid foods, and we call this process phagocytosis if we're dealing with solids. If we're dealing with fluids or liquids, we call it pinocytosis. So this is kind of like the cell drinking, whereas phagocytosis is the cell eating. So another example of phagocytosis here on the right-hand side. In this case, we have a very large particle. The plasma membrane is able to change shape and fold up and around our solid particle, or a very large particle. And again, these two sides of our plasma membrane will come together and meld, rehealing itself, the exterior of the plasma membrane, and creating a vacuole or a layer around our large particle. And that vacuole can then be used to break down that large particle if it is food for the cell, or if it is if the large particle is destined to be broken down and destroyed by the cell, as we see with white blood cells destroying bacteria that may be invading the body. So cytosis is a mechanism where the cell is able to take in large amounts of material into the cell, but it's also able to remove or secrete substances from the cell. And this is what we call exocytosis. So the first type is endocytosis, meaning going into, and we have exocytosis, exiting the cell. This is a process where substances are removed from the cell. So it might be removing waste products, or if they are secretory cells, they might be excreting hormones or neurotransmitters, chemical signalers for the body. On the inside of our cell, we have our substances, whether they be hormones, waste products, or neurotransmitters, are stored or packaged up into a vesicle. Okay, so again, this vesicle has a membrane around it, a lipid bilayer, which is the same as the lipid bilayer on the outside of the cell. These vesicles are then transported to the inner surface of the plasma membrane. So they're targeted there and they move to the inner surface of the plasma membrane, where the membrane around the vesicle will merge or meld with the membrane on the outside of the cell, with the cell membrane. When the vesicle membrane merges with the plasma membrane, it opens the vesicle out to the extracellular environment. So the vesicle membrane becomes a part of the plasma membrane. It opens up the vesicle, and whatever's stored inside the vesicle is then released to the outside environment. So this may be to rid the cell of waste products, or it might be excreting hormones or neurotransmitters or chemical messengers for the body. So here's another example NCEA question. This one is looking at active transport. So this was the second part of our question with our lugworm. This one is with the hog choker fish, which lives in estuaries where saltwater concentrations will change regularly. So again, we have a change in salinity, a change in saltwater concentration. However, instead of using passive transport, our hog choker actively adjusts the saltwater concentration of its body when it's in high salt concentration water. As the salt concentration increases, oxygen consumption also increases. So the fact that oxygen consumption increases, if you can remember your year 12 course, that might link in with, it might jog your memory, something to do with respiration, which we will be talking about soon. But let's have a look at the active transport involved. So if we explain how salt water moves across the membrane in the hog choker by active transport. So movement of our salt molecules across the cell membrane is via active transport. We need a carrier protein. Salt is moving against the concentration gradient from an area of low concentration inside of our cell to an area of high concentration outside of the cell. There are protein pumps in the cell membrane which are pumping those salt molecules out of the fish's cells and into the seawater. Because we're pumping against a concentration gradient, this process requires ATP or energy.
Just a question for you there, Amy.
Yes.
Have you ever had to explain cytosis specifically in NCEA, or do they not ask about that?
I believe I have seen questions with cytosis. A lot of these concepts or ideas around passive and active transport are generally linked with other life processes. So I have selectively chosen this question here, and we've focused on the active transport aspect of the question, but the rest of the question on the hog choker was linking it in with respiration, and we often see that questions around active and passive transport are linked in with ideas to do with respiration or energy use in the cell as well. Off the top of my head, I can't think of a question where cytosis was, like an explanation was required, but generally speaking, these sorts of questions around transport are often linked to other life processes as well. Does that kind of explain?
I think it does.
Yep.
Thank you. And a question from Ella. Is the answer to that question an achieved?
These were merit level.
Awesome. Thank you.
Thank you. All right. So these are some achieved level questions though. So in this case, we have brush border cells in the small intestine. When nutrients first enter the intestines, they move through the brush border cells by diffusion. So a couple of things to note. We see this odd shape to our cells. There's kind of cuboidal in shape. But they have these folds in the membrane up the top here. Okay, this is an example of animal cells taking on very specific shapes for the specific role they play. These cilia or shapes up here are designed to increase the surface area so that they can absorb nutrients through this process here and then moved into the cell. So these achieved level questions describe the process of diffusion. Okay, so in this case, we are talking about our nutrient molecules. Diffusion is the random movement of particles down a concentration gradient from an area of high concentration in the gut here to an area of lower concentration inside the cell. The process of active transport then is the movement of particles against a concentration gradient from an area of low concentration to an area of higher concentration. This process requires ATP. So your achieved level questions are generally going to be explaining a process or providing a definition for a process. Your merit level will be applying that to the question or explaining them in much more detail with reference to the context of the question as well. Let's have a look at proteins and enzymes. They're absolutely vital to the survival of any cell. Most cells, cells are made of mostly different types of proteins. So there are different types of proteins, and some of these are structural proteins or antibodies. signaling proteins, and enzymes. Our protein, we covered off a lot in the gene expression topic, but just to jog your memory, they're long chains of amino acids that are then folded up into very specific shapes. And they need to be in very specific shapes because that shape determines the function of that particular protein. They're held in the right shape by chemical bonds between the amino acids in that sequence, usually hydrogen bonds and disulfide bonds. And those bonds are really important to hold that protein in its correct shape and conformation. That shape and structure of the proteins is vital to their correct function. So on the left-hand side, we have an example of an enzyme. We can see our substrate, our circle, and our triangle are fitting nicely into the active site of our enzyme. If our enzyme were to change shape, our substrate wouldn't be able to fit in here. Okay, so the shape of our enzyme is vital to its function. On the other hand, on the right-hand side, we have our protein pump. A couple of different things, a couple of different ways we can think about it. If our protein pump were to change shape on the inside of this panel, our sodium and potassium wouldn't be able to bind to it. But also, if our protein were, 2/3 shorter, cut off here, it's not going to pass all the way through the plasma membrane. So that would also affect its function. So the shape of and structure of proteins is vital to their function. It determines correct functioning of the protein. Enzymes are one particular class of proteins, and their role is to catalyze chemical reactions. So they're able to speed up those chemical reactions by lowering the activation energy. They don't make reactions happen that wouldn't otherwise happen. They just speed them up. So enzymes are unchanged by the reaction. They don't take part in the reaction. They just catalyze it. This means that that an enzyme can be used many, many times. Okay, so it's not a case of an enzyme taking part in the reaction and then being turned into something else. An enzyme will reset and can be used for the same reaction many times. The important thing is that enzymes are very, very specific. Okay, one enzyme usually carries out one reaction. Okay, it will bind one substrate, perform one reaction, and produce one type of product. And that comes down to the shape of our enzymes too, which we'll look at shortly. If you're not sure if you're dealing with an enzyme, just have a look at its name. So most enzymes will end in the suffix "ase" or "ase", and they're often named for the reactions that they catalyze. So, for example, we have lipase, which is involved in breaking down lipids or fats. We have protease. Again, we've got that ase, and the prote part reminds us that it's for breaking down proteins. Lactase breaks down lactose. DNA polymerase, you've probably heard of, is involved in putting together the DNA polymer. So we have the ase part, which reminds us that it's an enzyme, and the first part usually tells us a little bit about the reaction that it catalyzes. On the right-hand side here, we can see the effect of an enzyme. So with our red line, this would be the amount of activation energy required up here without the enzyme for our reaction to proceed. Okay, with an enzyme involved, it is able to catalyze that process, which lowers the activation energy. This means that our reactants don't need to have as much energy to react and go on and form their product. So I said that enzymes were very, very specific. Okay, the reason they are very specific is they have a very specific active site. So an active site is an area on our enzyme, a groove or a nook in the protein structure that the enzyme's specific substrate can fit into. Now, the substrate is the substance that the enzyme is going to act on to catalyze a reaction. So it might be breaking down one substrate into several different products, or it might be binding several products and bringing them together to create a new product. So according to the lock and key model, this is where the substrate fits very precisely and perfectly into our enzyme's active site. So the active site is a perfect match for the substrate, and this is where having the correct shape or conformation of our enzyme is important. The substrates, once they're bound to the active site, are held in place by hydrogen bonds, and then our enzyme is able to catalyse the reaction. Once it has catalysed the reaction, our products are released here. If there is any change to the shape of our enzyme, it will affect the shape of the enzyme's active site, which will mean that our substrates are not able to bind and our reaction cannot proceed. Another model you've probably heard of, and hopefully my little animation works, is the induced fit model. So according to this model, the enzyme's active site will loosely fit the specific substrate. It'll be a reasonable fit, but when the substrate binds to the enzyme's active site then changes shape slightly so that it will better fit the substrate and lock it in place. This will also, or can cause a change in the shape of the substrate too. So it might put the molecule under a little bit of tension, which might again lower the activation energy and make a successful reaction more likely. Hopefully my little animation will work again. Oh, it won't. No. But we see the substrate come in, it binds to our active site. It's not a perfect match, but what we see is that the enzyme's active site then folds or changes shape around our substrate to match it exactly, puts the substrate's structure under a little bit of tension, which leads to a successful reaction occurring more likely. All right, let's talk about denaturation. So denaturation is important for enzymes. It's when the protein structure or shape has changed so that it no longer functions, no longer works. So remembering that proteins are, their function is determined by their shape. If we change the shape, that's going to have a massive effect on their function. So proteins are held in their structures by our hydrogen and disulfide bonds. So on the left-hand side here, we have our folded protein. We see this ribbon shape represents the amino acid sequence, and that sequence is folded up and twirled up on itself, and it's being held in that shape by hydrogen bonds and disulfide bonds. Because chemical bonds are holding our protein together, factors like temperature and changes in pH can affect those chemical bonds. So if we have a very high temperature, it can break those hydrogen bonds. If we have changes in pH, it can also affect those bonds as well. If those bonds are broken, this will lead to the protein losing or changing its shape. Okay, and for our protein, if we lose or change the shape of our protein, we will see a loss or change in its function. Okay, lots of cooking processes are involved or are causing denaturation of the proteins in the meats and the foods that we eat. So if you think about an egg white, when it comes out of the egg, it's a clear yellow type jelly-like solution, and if we heat that up, it becomes that firm white egg white that we like to eat, and that is a result of denaturation. The proteins inside the egg albumin have been affected. The bonds holding those proteins together have been broken, and we get these unfolded proteins which are no longer able to function, no longer to hold their shape. So there are a number of factors that affect enzyme function. We've talked about temperature. Okay, we know that if the protein gets too hot, the enzyme will denature, it will change its shape, it will lose its shape. This means that the enzyme cannot catalyse the reaction. However, if it's too cold, the rate of enzyme function will be low as well. Okay, and if you think back to year 11 acids and bases, this is to do with the collision theory in particles. We know that substrate particles that are too cold will move slowly. They won't have a lot of energy, so they won't be colliding very often with our enzyme's active site. Okay, this means that enzyme function will be low. Enzymes function at an optimum temperature. This is the temperature at which the substrate particles are able to collide more often. The reaction is able to be catalysed efficiently because our substrate particles have sufficient energy for the reaction to proceed. pH we've talked about, so we know that our proteins and enzymes require an optimum pH. If they're inside a cell, usually their optimum pH is about pH 7, which is neutral. If we have a change in pH, higher or lower, so a more acidic or basic environment, this can affect the way the substrate binds to the enzyme because it's controlled by hydrogen bonds, or it can cause the enzyme to denature. Concentration of substrates is also really important. Okay, so if we have a high concentration of substrates, we're going to have more successful collisions between our substrate and the enzyme active site. This means that the rate of reaction will increase. More collisions with the enzyme's active site means more collisions, more successful collisions, and a higher rate of enzyme reaction. And then lastly is inhibitors. So inhibitors are poisons or substances that prevent enzymes from being able to catalyze a reaction. When we talk about inhibitors, there are two types. There are site-competitive inhibitors and non-site-competitive inhibitors. Some inhibitors, they might be protein-based, they might be different chemicals or molecules, things like heavy metals such as lead, mercury, and cadmium are different types of inhibitors. With a site-competitive inhibitor, a site-competitive inhibitor is able to bind to the active site of the enzyme. So they usually have a shape which is very similar to the natural substrate of our enzyme. The enzyme can't tell the difference, so that site-competitive inhibitor is able to pop into the active site of our enzyme and sit there and bind to it. And this blocks the active site so the substrate cannot bind to it. Okay, it doesn't matter how much substrate you've got, if they are all competing for the same spot on the enzyme, the substrate is not able to bind. And if the substrate can't bind, the reaction can't proceed. It's like a car in a parking spot, if you like. So if this is our normal substrate binding to the active site on our enzyme, our competitive inhibitor is like our neighbour's car parking in our parking spot, which means that we're not able to park there. Our enzyme isn't able to bind its substrate, therefore the reaction can't proceed.
Got a good question from Thomas there, Amy. How do enzymes in the stomach not denature then?
They have their optimum pH is they are adapted and evolved to exist at very low pHs. So I didn't specifically talk about that example, but yes, when we talk about enzymes in a cell having an optimum pH, I did say that for most cells that's around pH 7 for neutral. For enzymes that are in our stomach cells, of course, their optimum pH that they operate at most efficiently is going to be much, much lower, so around about a pH 1 to 2. This means that if they encountered an environment or a situation where the pH was higher at around pH 7 or 8, then that would affect the chemical bonds between the amino acids and the protein holding the enzyme together, and you'd likely see denaturation of those proteins when they are at a non-optimal pH.
Awesome, thank you.
Yep. Okay, so the other type of inhibitors we've got is non-site competitive inhibitors. Okay, they do bind to the enzyme, but instead of binding in the active site, they bind at a location or a site away from the active site. So you can see here with our green enzyme, our normal substrate is able to bind to our active site. However, if we have a non-competitive inhibitor, which binds to a site away from the active site, it causes a change in the shape of our enzyme. Okay, it's specifically a change to the shape of the active site here. What this means is that our substrate can't bind, or binding is impaired. It's not able to bind the way it needs to, which means that our reaction also can't proceed. So the way that our non-competitive inhibitor works is it causes a change to the shape of our enzyme and our enzyme's active site so that the active site no longer fits its specific substrate. And if the substrate can't bind to the active site, our reaction can't proceed. But enzymes and enzyme function are vital for the cell to carry out cellular processes. And almost everything that the cell does is controlled by enzymes. They're super important. When we talk about DNA replication and the process that happens there, we're talking about enzymes. Okay, it is controlled by enzymes. When we look at photosynthesis and the process that happens there, it's controlled by enzymes. They're doing all the work. In respiration, there are many enzymes involved in that process. Okay, and lots of times when you get an exam question about enzymes, they will be linking it into a life process such as DNA replication or photosynthesis or respiration. So the enzyme topic is really important to get your head around, because it helps you answer those questions about DNA replication and photosynthesis and respiration, those other life processes, with greater depth and understanding. All right, so here's an example. The eastern oyster's cellular respiration and enzyme activity are affected by temperature, oxygen concentration, and cadmium, which is a heavy metal. Discuss how these factors can affect cellular respiration and enzyme activity in the eastern oyster. You should first describe the purpose of an enzyme, explain how temperature and cadmium affect enzyme activity, discuss how environmental temperature, oxygen concentration, and cadmium can affect the rate of cellular respiration in the eastern oyster. So again, because we haven't talked about respiration, I've mostly looked at when I've chosen this question as an example, we've looked at those questions around the enzymes. So the first one, a nice, easy, achieved, is describe the purpose of an enzyme. And it's a biological catalyst that speeds up a chemical reaction or which lowers the activation energy of the reaction. So nice, easy one there. Provide the definition. The next part is an explain. So we are taking this idea and explaining the effect of temperature on enzymes and the effect of cadmium on enzymes. A little bit tricky. They haven't specifically said much about cadmium, but they have said that it is a heavy metal. And if you remember, heavy metals are inhibitors of enzyme activity. So we're going to be talking about the effect of temperature on an enzyme and the effect of an inhibitor. All right. Now, sorry, this is a bit wordy. So respiration is a cellular process controlled by enzymes. Okay, and they function at an optimum temperature. If the temperature is too low, collisions between our substrate and the enzyme's active site will be less frequent. So the rate of respiration will be low and the rate of reaction will be low. As the environmental temperature increases, those substrate particles will be moving around faster and with more energy. So there will be more frequent collisions with the enzyme's active site. So respiration will also increase. We'll see an increase in the rate of reaction. Enzymes have an optimum temperature. The optimum temperature for our oyster will be the temperature at which the peak rate of respiration occurs, so where respiration is highest. Above this temperature, the enzymes may become denatured, which makes them inactive, and denaturation is irreversible. What it means is that denaturation will cause a change to the chemical bonds holding our enzyme together, and our enzyme may change or lose its shape, which will result in a loss of function. Enzymes are denatured because of a change in the shape and structure of the active site. It means that they can no longer fit the substrates involved in this process. If the substrate can no longer bind to the active site because the active site has changed, the rate of cell respiration will decrease or may stop altogether. So this has kind of linked together the effect of temperature with the rate of cellular respiration. So it's sort of both of those two bottom bullet points. Now to address cadmium, hopefully, there we go. So cadmium is a heavy metal and it acts as an enzyme inhibitor. It can bind to the enzyme's active site, blocking it or changing the shape of the active site. This prevents the substrate for the enzyme from attaching to the active site. If the substrate cannot bind to the active site, then the reaction cannot be catalyzed, therefore a product cannot be formed. So this prevents the enzyme from functioning correctly. All right, so let's move on to another enzyme-mediated process. We're going to look at respiration. So the site of aerobic respiration is the mitochondria. So let's have a look at the mitochondrial structure first. Here we have it on the right-hand side. They're often described as being a bit sausage-shaped, and they do have a reasonably simple structure, but it is very important to mitochondrial functioning. So we have our outer membrane. Okay, this contains the mitochondria and regulates the flow of molecules entering and exiting the mitochondria. So everything that flows in or out of the mitochondria has to go through that outer membrane first. Within the mitochondria, we also have this inner membrane here, and it is folded up into these wrinkles or these folds known as cristae. The whole purpose of this is that it increases the surface area for the respiration reactions to occur on. So many of our respiration reactions occur on this folded inner membrane. The cristae is the site of our electron transfer chain reactions. So they're very, very important. And inside our cristae, or inside our inner membrane, is this jelly-like filling known as the matrix. And this jelly-like filling contains many, many, many enzymes which are responsible for the Krebs cycle, which is another part of our aerobic respiration. So when we're talking about aerobic respiration, this is the process by which glucose is broken down to water and carbon dioxide, and in the process produces lots and lots of ATP. So the mitochondria's whole purpose is to produce ATP, which is used by the cells for energy, for driving lots and lots of different processes. We've looked at active transport already. ATP is involved in protein synthesis. It's involved in movement as well, so muscle fibers, flagella, which are like little tails on some different types of cells, phagocytosis, which we've already talked about. So cells need a lot of ATP. Cells with a very high energy demand, so things like muscle cells or cells that are producing lots of stuff and undergoing lots of exocytosis or secreting lots of substances, means that they will need lots and lots of ATP. And what we find is that cells that have a high energy demand have many more mitochondria than cells with a low energy demand. So you can have up to, around about up to 1,000 mitochondria in some cells that have a very high energy demand, such as muscle cells. Sperm have a very high energy demand because they are swimmers. Okay? They need the energy from movement to swim. So that's why we have included them there as well. What we also see that in these cells that have a very, very high energy demand, as well as having lots of mitochondria, the mitochondria that they do have many, many more folds in their cristae. So instead of having a few, they have many, many more folds, which increases the surface area available for all of those respiration reactions and ATP production. So you can see in my diagrams here, this is just a picture of mitochondria underneath a scanning electron microscope, and you can see those cristae are these folds that you can see, remembering that this is a cross-section, so it's as if we have cut through our mitochondria and had a look on the insides. Down the bottom here, I know it's very, very small, you can't really see, but you can see this must be a cell with a very high energy demand because it is just jam-packed with mitochondria. And if you can, I will make these slides available so that you can zoom in. But when you zoom in, you'll be able to see that those mitochondria are just absolutely packed with cristae as well. So they have a very large inner membrane, which is folded up into many more cristae, which allows for greater surface area for reaction. So aerobic respiration involves combining glucose and oxygen and producing carbon dioxide and water as our waste products. But the main point is that it produces lots of ATP and some heat energy as well. The name aerobic tells you that it requires oxygen. So oxygen is needed for this process to help break down the glucose. There are three parts to aerobic respiration. Glycolysis, the Krebs cycle, and the electron transfer chain. And it starts in the cytoplasm, but most of it occurs in the mitochondria. So we have glycolysis here in the cytoplasm or the cytosol. This is where glucose is broken down to pyruvate. The pyruvate then enters the mitochondria, where it passes through the Krebs cycle and then onto the electron transport chain. But I've got another slide with a bit more detail. So, as I said, glycolysis occurs in the cytoplasm. This is where a molecule of glucose is then broken down to pyruvate. And in the process, 2 ATP molecules are produced. So for every molecule of glucose, 2 ATP molecules are produced. That pyruvate is then moved into the mitochondria. Okay, so it goes through the Krebs cycle. This occurs in the matrix of the mitochondria, the jelly-like filling, if you like, which is packed with enzymes. Through a very complex series of enzyme reactions, those pyruvate molecules are then broken down to produce hydrogen atoms, which is the useful part, and carbon dioxide. And the carbon dioxide is waste. So our carbon dioxide waste products are released from the mitochondria and then later from the cell. But the hydrogen is what our mitochondria are looking for. Those hydrogen atoms are then passed on to the cristae, that folded membrane inside our mitochondria. Those hydrogen atoms passed on to the cristae then go through the electron transport chain. So the first thing that happens is that those hydrogen atoms are then ionized to H+, or hydrogen ions, by removing their electrons. So it's called an electron transport chain because the electrons are the important part. Once those electrons have been removed from the hydrogen, have quite a lot of energy, and they're passed backwards and forwards. Again, a very complex series of enzyme reactions. They're passed backwards and forwards between molecules on the cristae, and as they are doing that, what they're doing is they are using the energy from those from the electrons to turn ADP, or adenosine diphosphate, back into ATP. Now, if you remember, we thought of ADP as being our flat battery. So those electrons, and being passed backwards and forwards through those reactions in the electron transport chain, are recharging those ADP batteries and turning them back into ATP, adenosine triphosphate. And this whole process produces 36 ATP for every molecule of pyruvate. So it's very, very efficient. As a waste product, we see water produced. So once those hydrogen ions have done their job, they are combined with oxygen to produce water as a waste product, which again is released from the mitochondria in the cell. So the key points for aerobic respiration are that it requires oxygen. There are 3 stages: glycolysis, the Krebs cycle, and the electron transport chain. And as a result, there are 38 molecules of ATP produced for every 1 glucose molecule. In contrast, we have anaerobic respiration. Okay, so this takes place in the cytoplasm of the cell, not in the mitochondria. So the mitochondria are not required for this process. Compared to aerobic respiration, it is fast, but it's not very efficient. It doesn't require any oxygen, so it does have its purposes, but for every glucose molecule, it only produces 2 ATP, so not very efficient. In animals, we have the glycolysis reaction occurring in the cytoplasm, where glucose is turned into pyruvate. Again, this generates 2 molecules of ATP. As a result, we have lactic acid produced. As a result, pyruvate is then broken down to lactic acid. And we've all heard of lactic acid. It's the metabolite, or the compound that builds up in our muscles when we're under intense physical activity, and that's as a result of our muscle cells not being able to produce enough ATP from aerobic respiration. So they are switching into anaerobic respiration because it is able to produce ATP faster, even though it doesn't produce as much. But the downside is lactic acid is produced as a result. Slightly different situation in plants. Instead of producing lactic acid, our pyruvate is then used to make ethanol and carbon dioxide. So as I said, respiration is an enzyme-mediated process. Enzymes are responsible for all of the reactions involved in respiration. So all of the things that affect enzyme function in general will also affect the rate of respiration. So again, if it's too cold, it's going to affect the rate of those enzyme reactions, so respiration will be slow. If it's too hot, the enzymes may denature, in which case those respiration reactions can't occur, so respiration will stop. And organisms will have an optimum temperature, and this is the temperature at which respiration is happening efficiently at the ideal rate. Because it's an enzyme-mediated process, if the cells undergoing respiration are exposed to changes in pH, this can result in denaturation of the enzymes, which can halt respiration. What is important is we think about the concentration of the substrates. So we know that this affects enzyme-mediated reactants. And in the case of respiration, we're talking about glucose. And if we're talking about aerobic respiration, we're also talking about oxygen. So if our supply of glucose is low, then respiration will be slow, regardless of whether it's aerobic or anaerobic respiration. If we don't have enough glucose, respiration will be slow. And if we're looking at aerobic respiration, we also need a good steady supply of oxygen as well. So if oxygen supply is limited, then this will also slow respiration. Inhibitors, there are inhibitors that prevent enzymes involved in respiration, so this would affect the reaction. But also crucially is demand. So the demand for energy. And if animals are undergoing activities that require a lot of energy, so they have a very high demand for energy, such as running or flying, this will cause an increase in respiration in their cells. So their cells are able to increase the rate of respiration to provide more ATP for the cells to undergo those or carry out those activities that our animal needs. This only works up to a maximum though, so once the animal has reached its maximum rate of respiration, it's not able to continue to increase the rate of respiration beyond its maximum. All right, let's have a look at chloroplasts. These are how plants make glucose. So for animals, because they are heterotrophs, they eat all of their nutrition or drink it. They're able to take in glucose or to digest their food and produce glucose as a result of digestion, and then the glucose goes on to enter the mitochondria and undergo respiration to provide energy. However, plants don't eat as such. They need to make their own food. And the way they do this, or their food, is glucose, and they produce glucose through the process of photosynthesis. So for photosynthesis to occur, we need a chloroplast, which is the big green blob on the right of the screen. Reasonably simple structure again, but again, that structure is very important to the role that our chloroplast plays. So we have our outer membrane. Again, this contains the chloroplast and all of the vital parts that make it up, but also controls the flow of substances into or out of the chloroplast. We have an inner membrane which encloses the stroma. The stroma is the clear jelly-like liquid which is inside the chloroplast. It's clear so that light can pass through to the thylakoid, okay, which is very, very important. So thylakoids are these stacks here. So they are stacks of grana or granum. Granum are green. They are green because they contain chlorophyll, which is a pigment that is able to absorb light energy. The light energy is then used in the photosynthesis reactions. The grana are arranged in these big stacks, which are then linked together. Those stacks are known as thylakoids. And the whole purpose of the structure is to, once again, maximize the surface area available for the reactions. In this case, the light-dependent reactions. All right. The energy from the sun is used to convert carbon dioxide carbon dioxide and water into glucose. The plant then uses the glucose for respiration, and oxygen is produced as a waste product. So we know that plants take in carbon dioxide, and I'm sure we're all familiar that plants need water as well. Chlorophyll, that green pigment, is able to absorb light energy and is able to split water, and we get carbon dioxide and water being used to create glucose, and oxygen is our waste product. There are two main chemical reactions involved in photosynthesis, the light-dependent reaction and the light-independent reaction. So the light-dependent reaction, from the name, tells you that it depends on light. It occurs during the daytime, whereas the light-independent reaction does not require light and can happen at any time. So let's start with our light-dependent reaction. This takes place on the grana. Those are the green parts, the parts of the plant, or through the parts of the chloroplast that contain chlorophyll. The chlorophyll pigment absorbs light energy from the sun, which excites electrons in the thylakoid membrane. Okay, remember that membrane, the thylakoid, the stacks of grana has lots and lots of surface area. So those electrons become highly excited, and they are passed backwards and forwards through those membranes again and used to make ATP from ADP. So again, we're recharging those flat ADP batteries and charging them up to be ATP. This energy is used to split water, a very difficult process, but we split it up into hydrogen atoms and oxygen gas. So the oxygen gas is a waste product at this point, and it's released from our chloroplast, but the hydrogen is the important part. The hydrogen is then picked up by a carrier molecule called NADP, which has a very long name that you don't need to know. But the NADP, once it has attached hydrogen, becomes NADPH, the H for the hydrogen that is now attached to the NADP. That carrier molecule then transfers that hydrogen into the stroma of the chloroplast. So the stroma is clear to allow light to pass through it, so the light can hit the chlorophyll on the grana. The stroma doesn't need to have any particular colour, but it is absolutely teeming with enzymes. So the light-independent reaction occurs in the stroma. This is where carbon dioxide and hydrogen, transported by the NADPH, again enters a very complex series of reactions called the Calvin cycle. Many steps to the Calvin cycle, but the result is that glucose is produced as a result. So the light-independent reaction requires the carbon dioxide that plants take in, and they combine it with the hydrogen from the water. ATP used in the light-dependent reaction is used to run this, created in the light-dependent reaction is used to run this part of the cycle. So it runs the light-independent reaction. So the carbon dioxide and hydrogen are combined to create our glucose, which is our C6H12O6. So again, photosynthesis, many enzymes involved in this process. Okay, this means that all of the things that affect enzyme functions will also affect photosynthesis. And we've talked about temperature, we've talked about pH, and we've talked about inhibitors. So all of those things will affect the rate of photosynthesis. So if the temperature is too cold, the rate of photosynthesis will be slow. If the temperature is so hot that it is denaturing the enzymes involved, then photosynthesis will stop. More importantly is the concentration of substrates. So again, if we have a shortage of carbon dioxide, then our light-independent reaction can't proceed. If we have a shortage of water, then the light-dependent reaction can't proceed. And we see a slowing or a halt of photosynthesis as a result. Okay, so we need to have an adequate supply of both carbon dioxide and water for photosynthesis. The next one is light intensity. So we know that the process of photosynthesis requires energy from the sun to be absorbed by the chlorophyll pigments. If there is a low amount of energy or low amount of intensity of light coming from the sun, then there is going to be a low amount of energy absorbed by our chlorophyll. If we increase light intensity or increase the amount of energy that our chlorophyll is able to absorb, this will increase photosynthesis. But again, it's up to a maximum point. So there will be a maximum point at which photosynthesis could occur, beyond which you can increase the light intensity all you like, but the rate of photosynthesis is not going to increase beyond that point. Okay. Mitosis. Mitosis is a type of cell division. We know that its purpose is to produce daughter cells that are identical copies of our parent cell. These cells are used for growth and repair of tissues. So they need to be exact copies of the parent cell because they are replacing dead or damaged cells, or they are increasing the number of those cells by through growth. Again, this is a process not quite so intrinsically linked to enzyme activity, but certainly one of the first stages or the first things that needs to happen before mitosis is DNA replication. So as we walk through these slides, or as I walk through the process, we've got mother cell here, our parent cell, and the first thing that happens is we see replication of the DNA. And we have these X-shaped chromosomes here. So those X-shaped chromosomes represent two copies of the chromosome attached in the middle by the centromere. So those copies, when they are joined together like this, are called chromatids. During meiosis, what next happens is that those chromosomes line up down the centre of the cell, and the chromatids are then pulled apart by the spindle fibres, creating two new daughter cells. And these new daughter cells are entirely identical to one another, and they are identical to the mother cell as well. So there's no crossing over involved, there's no recombination. They are identical copies of the mother cell. So there's a few things that affect mitosis. Okay, so the rate of mitosis is high in organisms that are undergoing growth. Okay, so whether it be a plant or an animal, if they are undergoing a period of growth, for animals, we're talking about young or embryonic animals. For plants, looking at saplings, if they're undergoing a lot of growth, then there is going to be a lot of mitosis occurring. They need those extra cells to grow. If our plant or animal is undergoing a stage of life that requires repair of cells, either due to injury or damage to the tissues, then there's going to be a lot of mitosis occurring to create cells to replace those damaged cells. The availability of nutrients and energy is also really important. So we know that substances and nutrients are required to build new organelles, to build new cell components, which are all needed to be duplicated when our cells replicate. So replication is slowed down if there is insufficient amounts of nutrients or energy available to the cell. It means that they're not able to go through replication or mitosis as quickly as they could. Some cells will, or based on the location, will have higher rates of mitosis than others. So this is again where there is periods of growth happening. So if we're talking about a plant, areas of the plant where you will see high rates of mitosis are, of course, in the shoots, the growing shoots of the plant, and also the roots or the root tips. There's a lot of mitosis occurring there because those parts of the plant are continually growing. They are replacing cells. Another example of where location is important is cells in the gut or the intestine of an animal. So because there is food and material passing through the intestine constantly, that provides friction and wear against the walls of the intestine. This will damage the cells and rub them off. So the cells in the wall of the intestine need to be undergoing a high rate of mitosis to be able to replace those cells that are rubbed off due to the food passing by. And another one is environmental factors. So mitosis and DNA replication is an enzyme-mediated process. So any factors that affect enzyme function, such as temperature and concentration of substrates, will also affect the rate of mitosis as well. So, I might come back to the slide, but we're going to have a look at DNA replication. DNA replication is the start of mitosis, which is why it's very, very important. And again, it is mediated by lots of enzymes. So just by looking at this diagram, you can see, you will hopefully identify things like DNA polymerase, helicase. There are a lot of enzymes involved in DNA replication. But let's have a look at the structure of DNA first.
Just a wee heads up, Amy, we do have another webinar starting at 4. We'll probably need a bit of lead-in time to set up for that one. I think we've gone slightly over here, but would you be able to wrap it up within 10 minutes?
Yes. Yep.
Cool. Thank you.
Thank you for the heads up. All right, so we know DNA is made of a series of nucleotides. We know it has nitrogen bases, a deoxyribose sugar, and a phosphate group. And DNA is made up of a long chain of these nucleotides. Each sugar is linked to a phosphate group, which is then linked to a phosphate, creating the phosphate sugar backbone of our DNA. And between our bases are hydrogen bonds, matching our bases up according to the complementary base pairing rules. Here we have a schematic of our DNA. It's made up of 2 strands, we know. We say that these strands are anti-parallel, and that refers to the backbone up the side of our DNA. So you can see that in this case, our sugars are pointing upwards, whereas on the other side, our sugars are pointing downwards. So we say that the top end with our sugar pointing upwards, this is our 5' end of our strand, and down the bottom here is our 3' strand. On the other side, so that the 2 strands can fit together nicely, they need to be running in opposite directions, or the other strand is effectively upside down. So you can see this pointy part of our sugar here, this is our 5' end, and down here is our 3' end. So this is what we say when we say these strands are anti-parallel. They run in opposite directions. Our 2 DNA strands together are held together by these hydrogen bonds, are our base pairs, forms a double helix structure. One strand, we know I've just said, runs 5' to 3', and the other is 3' to 5'. 4 different bases. We know that adenine only binds with thymine and guanine only binds with cytosine. This is our base pairing rule, and it is crucial to DNA replication. So the point of the base pairing rule and how it relates to DNA replication is it helps to ensure that we get an exact copy of the DNA that we are making. Because thymine will always bond with adenine and guanine will always bond with cytosine, we can always build an exact complementary copy if we only have one strand. We'll talk about that shortly. Here is our process. Okay, we have a double-stranded DNA here, part of our chromosome, and it is undergoing DNA replication. One of the first things that happen is that an enzyme, DNA helicase, starts or begins DNA replication by unzipping the double helix. Okay, so this is our double helix, and it is unzipped it. You can see in this portion here, the 2 strands are still held together by those hydrogen bonds. So the helicase breaks those hydrogen bonds and unzips the 2 sides of our DNA molecule. This creates what we call a replication fork. So here are the 2 tines of our fork. Single nucleotides line up against our DNA template strand. Okay, and DNA polymerase then links them together to form a new complementary strand. And you can see that that's what's happening there. These free-floating DNA nucleotides are being linked on by DNA polymerase and linking them against our template strand according to the base pairing rule. When DNA is replicated, the final helix is made up of 1 brand new strand and 1 old strand. This is what we call semi-conservative replication. So this is our old strand in blue. Okay, our new strand for the top one has been built in red, and it is an exact complementary copy to the blue one. The other strand of our old DNA molecule down here in blue, we have a new strand built alongside it, again according to the complementary base pairing rules. The effect is that the 2 halves of this DNA strand here have split. We have new halves formed for each of them, and these molecules, top and bottom, will be identical. So semi-conservative means half saved, if you like. So the half is the old part, has been saved, and we have built a new strand alongside it. Oh, where have my slides gone? Okay, let's go back. So here we have our replication fork. We've got DNA helicase is unzipped, and we have 2 strands. We have our leading strand here, okay, which is the top one, and we have what's called a lagging strand. And the reason for this is DNA polymerase. DNA polymerase operates in the 5' to 3' direction as it moves along the DNA strand. Okay, if you remember, because our DNA strand is anti-parallel, one strand is oriented from 5' to 3', and the other one is from 3' to 5'. Okay, so our DNA polymerase can quite happily move along one strand of our DNA in the 5' to 3' direction, linking on those nucleotides, binding them on, and creating a complementary strand to our template up here. The problem arises with our bottom strand down here, called the lagging strand, because DNA replication happens slowly along that strand, so that's why it's lagging behind. And the reason is that DNA polymerase can only move in this direction. This is a problem. So at the top here, you can see our leading strand. The arrow is pointing to the left. It is happily moving along the DNA strand, adding on nucleotides and building a new one, whereas in the lagging strand, it's not able to do that because it's moving in the wrong direction. The way it gets around this is by taking little jumps. Okay, so because this bottom strand is 3' down this end to 5' down this end, DNA polymerase can't move along in the 3' to 5' direction. So what it does is it jumps over a few nucleotides and sits down here at the top of our DNA strand, and it moves backwards, okay, in little chunks. We call those chunks Okazaki fragments. Okay, so our DNA polymerase is able to jump over nucleotides, work backwards, okay, until it meets up with the part that it's already done, then it will detach and jump over again. Okay, so it is, instead of moving continuously along the strand as it does for the leading strand, it is taking the lagging strand in small bite-sized jumps. Once DNA polymerase has formed those Okazaki fragments or jumps, we have another enzyme called DNA ligase, which comes along and links together all of those Okazaki fragments. So if we think about the name DNA ligase, ligase is like a ligature or a tying together, and the A's tells us that it is an enzyme. So DNA ligase is tying together the pieces of DNA, those Okazaki fragments. All right. So because DNA replication is an enzyme-mediated process, it's going to be affected by the same things as our other enzyme-mediated reactions. So temperature is going to be key as well. If temperature is too low, DNA replication will be occurring much, much slower than it should. If the temperature is too high, we will have denaturation of enzymes like the helicase and the DNA polymerase. So we're not going to be able to see that occurring. Those reactions are not going to be able to proceed. And substrate concentration is also key. So if there is a limitation in the number of these free nucleotides that are required to build our new DNA strands, then DNA replication will be slowed down as well. All right. At my 10 minutes. How are we doing, Doug?
Yeah. Is there anything else? You've got 2 minutes if you want to cover anything else.
No, I think that's good. I seem to have, some of my slides seem to have dropped off the end of the slideshow, which is regretful, but hopefully I've covered off everything. But if people have questions, now is your chance. You have 2 minutes. So let me know.
I think you've answered everything that's come up so far,
Okay.
but I will say very big thank you.
That's all right.
That's a mammoth job, boiling down cell processes like that, and you had some beautiful slides there. The animations worked almost perfectly for most of them, I think. It was maybe just that little one with that you identified that for whatever reason didn't work. So most of them seem to work really well.
Okay.
Thank you for all the time and effort that you've put into that.
That's all right.
We'll look forward to hearing from you on Monday at 2:00.
Yes.
And then we've got the last one, which I think is on Tuesday at 11 a.m. Is that correct?
That's right, yes. And I can make those slides available to you if other people want to look.
Yeah, if you don't mind, that would be brilliant. And I'll pop them in the description for everyone who wants to look back over those in the future.
Okay, fantastic.
Awesome. Thank you so
Cool.
much, Amy. Have a great
You're
weekend.
welcome.
Ka
Thank
kite.
you. Ka kite.